Hello everyone, welcome back to this cell signaling course. If you recollect, in the last class we discussed how different type of receptors are regulated at receptor level. Basically phosphorylation, dephosphorylation, desensitization, downregulation or binding with an inhibitor. And we saw multiple different types of receptor which we have studied until now, where engaging in one or other sort to have regulation on their activity in terms of their activation and engaging next set of molecule. What we understood until now is that all these receptors, whether they have integral enzymatic activity or they are dependent on another molecule which has an enzymatic activity, but they were not the direct molecule which were carrying out the signaling. Rather, they were recruiting next set of molecules which were either adapter, effector, enzymes and so on to mediate the signaling response. Today we are going to discuss the channel type of receptors, where basically receptor itself is carrying the signal forward and sending the output. It is not activating something as a next molecule, rather engaging in itself to mediate the signaling. So what we have discussed until now is that there are receptors with integral activity of signal transduction. We will be discussing that today. Here we have until now discussed the receptors which have enzymatic activity, but they do not have the signal transduction ability to themselves, like whether it was receptor histidine kinase, receptor serine threonine kinase, receptor like kinases in plant, or receptor tyrosine kinases. They needed another set of molecule for signal transduction. Whether it was a receptor which was on the cell surface, or even the ligand which was on the surface. In both scenario, what you notice is that next set of molecule recruitment in response to certain phosphorylation or receptor activation was required. So such receptor although had enzymatic activity in themselves, but they needed a next set of effector molecule for signal transduction. Then we discussed another set of receptor where enzymatic activity was not integral to the receptor, but they dependent on other molecule which carried the enzymatic activity. Like in case of JAK-STAT, if you recollect, JAK receptors, they bound with the JAK which phosphorylated the receptor, which brought the STAT molecule. The STAT was phosphorylated and this moves to the nucleus as dimer. Or we talk about these GPCRs, which were 7 transmembrane receptors. They first associated with a kind of G alpha. This G alpha activated next set of event, whether it was adenylyl cyclase activity, phosphodiesterase activity, or activity of phospholipase C beta family. In both the cases, receptor itself was not conducting the signal which is being conveyed at the cell surface. Then we have seen, or we have discussed in the very beginning, that when we discuss different types of receptor, and this is where we come to the other type of receptors which have integral signal transduction activity. What I mean by that is that these receptors have a conformation which seems to be closed, and these can be activated so that this conformation changes and it becomes more of an open type. Thus, the message which has been delivered through signal is conveyed through the receptor itself. In many cases, these are ion channels which basically allow influx or outflux of the ions. And most of the time they are like sodium, potassium, calcium type of ions which are there. So the receptor responds to a signal and allows the signal transduction to happen. In this particular case, it is the movement of certain ions, which we will be seeing in further slides. So these ion channels we are going to discuss more as receptor. As I told you, a receptor could be existing in the closed form, which is not allowing any movement of the ions, or it can exist in the open format where it will allow the passage of the molecule. Some channels could be very specific to which molecule, which ions they are allowing to pass through. Some channels will be non-specific. Some channels are very tightly regulated that when they open, and some channels are intermittently closing and opening, so they are kind of leaky channel. So we will see leaky channels, we will see ligand gated channels, we will see voltage gated channel and mechanically gated channel. What does it mean? That these channels do not have any direct control over there in terms of when they open and when they close. The second type of channels, they will be responding to presence of a ligand, and availability of that ligand opens the channel. So let's take some example. So here when we talk about randomly opening and closing channels, so this is the one which is leaky type. Basically in closed format it does not allow any ion movement. But when it opens randomly, it allows movement of ion. Like in this case, you see potassium ion is moving out. Such channels will be present on neuronal surface, which will be enabling that resting membrane potential formation. We know that resting membrane potential is around 70 millivolt negative. It is because more positively charged ions are accumulated on the outside of the plasma membrane or neuronal surface. The other type of channel which I mentioned earlier is ligand gated channel. This ligand could be anything. It could be a small molecule, it could be an amino acid, it could be some sugar or something like that, which opens the channel. So what you notice here is that channel is composed of multiple subunits and it has binding sites for these ligands. When the ligand is not available, it acquires a conformation which does not allow any passage. No passage allowed basically here. So when ligand is present, what will happen? This binding will induce a conformational change and you will see movement of ions. In this particular case, we are taking example of acetylcholine. When acetylcholine binds to its receptor, basically the extracellular surface, which has been the property of all receptor, that ligand binding domain is projected outside the membrane. So when acetylcholine binds here, a different conformation is acquired. Now it allows more calcium ions to move in or sodium ions to move in. What goes out is potassium ion. So in some sense, this movement of calcium and sodium inside allows the depolarization of the membrane. And if we recollect, when we were discussing a particular type of G alpha, that was G alpha T, which was helping in the visual perception, there you saw that the channel was ligand gated and was responding to cyclic GMP. When cyclic GMP was converted into GMP, it could not bind to the channel, thus the channel was closing. So there are so many ligand gated channel which allow the passage of ions or small molecule when they are open. So what you are noticing here is that receptor itself is engaged in conveying the message. It is not recruiting any next molecule to perpetuate the message or do the signal amplification or signal transduction. The third type of these receptor, before we go to that, let's discuss some of these acetylcholine receptors which are of two different types. What you just saw, as this receptor which had a conformation on which ligand was binding, and then the movement of calcium and sodium happened. This is called nicotinic acetylcholine receptor. This ensures that more calcium moves in or sodium moves in. There is another type of acetylcholine receptor which is called muscarinic acetylcholine receptor. It is important to put these two in perspective, because you will often see that you have lot of such receptors which are present on the neuronal surface. But you also have this kind of receptor for acetylcholine, which is called muscarinic acetylcholine receptor, and this is a GPCR type. So you know that when such a stuff is engaged, you will need G alpha. So second molecule. So receptor itself is not serving as a channel or signal transducer, rather it is dependent on that. And if you recollect one of the G alpha, which was G alpha Q, by breaking down PIP2 to DAG and IP3, and this IP3 was working on internal stores to release the calcium. So the response of increased calcium inside the cell surface is being, or increase in the calcium concentration inside the cell, whether it is nicotinic receptor or the muscarinic receptor, both are achieving that goal. However, the routes are going to be very different. The other type of channels, ion channels, which are serving as a receptor, are called mechanically gated channel, which means that if there is a stretch, there is a pull, there is a motion involved with respect to the cell, you will see these channels responding. So such channels are present in our skin and many other organs which are sensing the stretch or any mechanical changes. So normally this channel will remain closed in the absence of stretch, pull, pressure, whatever you say, something which is being sensed by the sensory receptors which are present in the skin. There these channels open when there is stretch pressure applied. So when these channels will open, you will see movement of molecule happening inside. Basically the movement of calcium, sodium and so on depolarizes the neurons on whose surface these channels are present. Basically this depolarization is an action potential which is an electrical impulse, and this continues through the axons and multiple neurons to different regions of the brain depending on which part of the skin has been, the stretch has been applied or pressure has been applied. So you saw there are channels which are leaky channel, there are channels which are ligand gated, then there are channels which are mechanically gated. Now we have another channel which are called voltage gated channel. So when you all would have read that how action potential is generated, there is a difference in the voltage which occurs at the cell surface, which may be initiating the opening and closing of the channel and activation of the and propagation of the action potential. So such channels sense the membrane potential difference and accordingly they work. They are present in all neurons. Normally when you have a resting membrane potential condition, so they have a unique provision where an extension of the channel blocks the entry of things, no entry. And whenever we are saying blocks the entry of certain molecules, we are talking about the ions. Now when there is a change in the potential, so from the previous step, if channel is present over here and the change in the potential is coming from step before, it will be sensed at this channel and the channel will open. What happens that the block which was there, something which was blocking the exit entry of ions, is now moved to a new conformation, and this opens the channel and allows things to move out and thus respond to these voltage differences and maintaining the voltage difference back to normal. So one more particular type of receptor which is in class of ligand gated channel, we saw for the acetylcholine a ligand gated channel. These are basically the channels or the receptors which are called NMDA receptors. These NMDA receptors, they are again having two units. If we consider unit N1 for NMDA receptor and subunit N2, they are present as a dimer. 2N2, 2N1 and so on makes a tetrameric unit. This tetrameric unit is going to be activated, and this activated channel will allow the conduction of ions. These channels which are these receptors which are in the class of NMDA, they are ligand gated because they will need certain molecules, and they are non-specific ion channel because they allow not one particular ion, rather a combination of ions. And the ligand here, this is a glutamate receptor. So basically it will respond to glutamate, NMDA, there is something called AMPA molecule, glutamate itself. So once the molecules bind over here, they will open the channel. What has also been understood in particularly in this case is that these glutamate channels or NMDA receptors, they are present in postsynaptic neurons. So if I draw a simple structure of a neuronal synapse, so this is presynaptic neuron and this is postsynaptic neuron. Basically these are neurons which are filled with neurotransmitter which is glutamate. When this glutamate is released, it should bind on the surface. So here you will have the NMDA receptors. Detailed study of this receptor suggested that they are kind of channels which detect the coincidence. Coincidence meaning other event of receptor binding to something. So these receptors, particularly if we discuss about the NMDA, and simply I draw like this as the tetrameric assembly, these receptors are regulated by presence of magnesium. It's basically blocking things here. It does not allow the ion movement. Magnesium is blocking it. When you have the ligands for this binding, some of the ligands I already told you, NMDA could be one, glutamate could be one. In addition, this binds to glycine or serine. So binding of all these molecules on this receptor may still not activate the channel. You need to dislodge this magnesium for the activity of this channel. So if in the vicinity another receptor is activated, which now helps in the removal of this magnesium, you will see ions moving in, sodium or calcium. So the occurrence of another event at the adjacent membrane is kind of indicating that, or NMDA receptor activation could also be assessed as occurrence of another event in the vicinity, the activation of a receptor in the vicinity which helped in the opening of such receptors. Now there are something called transient receptor potential channel, TRPV. These are very interesting kind of receptors. They are present in all our cells. They primarily mediate the perception of heat, cold, pain, stress and many more things, even related to taste. One of the things which this TRPV channel with respect to taste is capsicum. This is what is sensed by these channels. There are multiple of the classes of TRPV channel. They are present all over the body. Depending on the location, they will be participating in perception of heat, cold, pain, stress, and so on. And very recently, Dr. David Julius and Ardem Patapoutian received the Nobel Prize for their work on TRPV channel. So what are these TRPV channel? They are non-specific cation channel. What it means that they do not differentiate between the ions, whether it is potassium, sodium, or calcium. So when there is and these are basically ligand gated channel. You will need the ligand for them. So capsaicin, which is present in capsicum, is serving as a ligand here. These channels, when see the presence of the ligand, they open up and they allow the entry of calcium, magnesium, sodium, potassium. As I said, they are non-specific cation channels. So what basically happens to these channels? These are tetrameric channel. There are homo-tetrameric assemblies. So these 4 channels, if I say, they are oriented like this. Each subunit is represented as one bar here. So when and these are kind of embedded in the membrane, what we are looking at is the top view. And the projections from these channels close, or these individual subunits close the pore, so there is no movement of. When you have ligands do bind to 2 units. So basically for a tetrameric subunit, you have 2 ligands which are binding over here, and each one of them has a ligand binding position. So in this case, each ligand is dimeric. If the ligand is not dimeric, you will need 4 subunits of the ligand to activate the channel. So what will happen in these cases? These channels will be opening up, basically a new conformation you can think of, which will be coming up because of the, and even if those projections were there, they are moved aside because of the ligand binding, and this channel area which is generated in between, this will allow the movement of ions. So these movement of ion will basically, you think about the surfaces, cell surfaces where these things are being sensed. Largely these changes are being assessed by neuronal or cells which are directly in contact with the neuron, and this movement of ions basically activates those channels by depolarizing the neuron. And this depolarization, as I mentioned earlier, in form of some sort of electrical impulse is conveyed to the brain, where that electrical impulse is decoded and things are understood in a right context. Apart from multiple of these things which I spoke to you, there are certain pumps which are also present in the membrane. We know that there is something called sodium, potassium, ATPase pump. How these pumps are different from the channels which we have been discussing? Here, need of ATP is must. That ATP hydrolysis drives the movement of ions, where 3 sodium is pushed out and 2 potassium is brought back. And thus the difference between the charge that helps in the generation of resting membrane potential. So what you are realizing, that these channels or these receptors which we are calling so-called, whether they require an ATP or not, or they by ligand binding conformational changes, they induce the movement of ions. the direction of the movement of ion, the nature of the ion which is moving across, allows the depolarization or repolarization of the membrane or establishment of the potential across the membrane. So what we have understood in the class today is that these channels type receptors, these channels are type of receptors. They present a situation where receptor itself has the transduction ability, and this transduction ability is in terms of movement of ions. Then these channels, we know that they can be activated by many different routes, whether it is ligand binding, potential difference, or stretching, or the pressure application. We saw for acetylcholine, there is a receptor which is channel type and allowing the movement of ion directly, or it has a channel which is involving a GPCR which utilizes a next molecule which will be serving towards the signal transduction function. We saw example of NMDA receptor, which was a glutamate receptor, which was allowing the movement of ions inside, and it required binding of not just glutamate but also glycine or serine. More importantly, we saw that how it is helpful in understanding phenomena of coincidence, that another event of membrane channel opening and allowing the movement of ion can be detected just by seeing if the NMDA receptor is being activated. We lastly saw the TRPV channel. These channels are widespread in the body. They sense heat, pain, taste, and many more things, and they are non-specific type of cation channel, which means that they will be working for magnesium, calcium, sodium, and potassium. And in case of NMDA, we saw how magnesium was inhibiting or some sense guarding the channel. Thank you.